La emisión está comenzando. Todos los asistentes están en modo de solo escucha.
Materia subir. You have to turn on this. Yeah.
Da me c'è un rumore che c'è veramente qualcuno
And
che parla costante.
shade this one.
Io non sento nessuno, tra l'altro turn off mic. Tra l'altro non so se tu mi vedi cioè il video, vedi, cioè lo program, for example. All attendees organizing. Questions? Sì? Imma, you can hear me. Okay, I can. There's too much noise in my. Okay, now it's better. Okay, maybe we can start then. Wow, there is too much noise here. Someone is still on. I don't know how to unmute. Maybe somebody can help me in.
Hi, Gabi.
Hi, Carla.
Hi.
Is you?
Yes, it's me.
Maybe from you. There's a big noise in my headphone.
No,
I can hear a background noise.
I just
Okay.
unmute myself. I was muted, and everyone else is muted as well.
Okay, then it's only me then. Just wait a minute, and we will start soon.
Okay.
Just 1 minute. Okay. Okay, I think here we are. Yeah, it's much better now. Thank you, Carla. So welcome today on our seminar series on extracellular vesicles 2020, Napoli, from membrane biophysics to drug delivery. Today is, I try to go ahead, but not, just wait a moment. Okay, maybe we will do it like this one because it's not going ahead. So today, unfortunately, we are available only online, so we are not gathering in Napoli at the CNR as usual. So it is due to because of the coronavirus emergency, what we have here in Italy. You can, and I think many of you will join online. So all those who cannot come still can register online just using the registration page, what you can see here, but I also sent it over by email. I just checked a few in this morning, and actually we reached something like 92 registration. So we are getting closer to the 100, the magic 100 number. So to all who are not, who are joining us for for the first time. I just wanted to say what are our aims. So we basically want to bring together researchers who are interested in extracellular vesicles. We meet every Wednesday at 12 p.m. In Napoli, generally at the CNR, today only online. The main aim of this initiative is to promote collaboration in Campania region. We promote all the studies of all types of vesicles, small one, large one, plant-derived vesicles, hydro-derived vesicles, or even blood or human extracellular vesicle research. We would like to give some introduction or some lecture on all type of sciences. So we promote basic science to applications, and with this initiative we would like to also give MPS space for discussion. This seminar series is sponsored by the Italian Society of Extracellular Vesicles, called EBITA, and also the VES4US European Horizon project. We also try to make at least one tweet a day at the seminar day. So today's tweet is on nanoTOM, because today we have this fourth presentation from Ramesh Bocca, and he's going to give some insight on tomato-derived vesicles. The previous presentation was from Chiara Ciardiello, and this is also to take the opportunity to say many thanks to Chiara for the very nice presentation, which was on human extracellular vesicles, the big ones, and when she showed last week us that how size matters in tumor progression. So I don't want to take a lot more time. So today, after this small introduction and also the presentation of Ramesh, he will talk about half an hour, and then we can discuss. We will see whether we can open the microphones and discuss it live, or you can just type some questions, and I can or Ramesh can see it to answer. So we will see how it works. Unfortunately, the pizza is not for today, but we hope that next time we can go out also for pizza together with those who are going to join. So just a few. Today's speaker is Ramesh Bocca. He's a postdoc in the EVS lab of IBBR CNR in Naples. Ramesh did his PhD, his BSc in 2005 in mathematics, physics, and chemistry in Kakatiya University of India. 2007, he received his MSc degree in organic chemistry from the Osmania University, Hyderabad, India. And he received his PhD in 2015 from CSIR, Indian Institute of Chemical Technology, also in Hyderabad, India. Ramesh started to work in 2008 when he was first project assistant, then junior assistant, and he got also the senior research fellow position at the analytical chemistry division of the CSIR IICT Hyderabad, India. Then in 2015 he changed his career development plan, and he decided to go for industry. So he spent a couple of years at Eurigene laboratory. Eurigene is a company, actually. It's a drug discovery company, very important drug discovery company in India, based in Bangalore, Hyderabad. This company is a fully integrated drug discovery infrastructure, and they do drug discovery from heat generation to preclinical development. So Ramesh was a scientist at the medicinal chemistry department at Eurigen before decided to come to Italy. So in 2018, Ramesh moved to Napoli, moved to my laboratory, where he was awarded for a Marie Curie Sklodowska Fellowship. He started this adventure at IBBR CNR in EV research, and actually he now works at the extracellular vesicles and mass spectrometry group on the development of tomato-derived vesicles for nanovectors. So with this one, I will try to give the word to Ramesh, and thank you very much for your attention. So if we can just unmute Ramesh.
He's already on mute. So Ramesh,
He's already unmuted. Okay. Okay, thank you.
Gabriel.
Can you hear Ramesh?
Yes, Gabi, but I want to show my slide.
You can hear Ramesh. I think we cannot hear him very well, so I will ask him to join the seminar from my computer, because as I told you, he's so he's in my lab. So I will open his. Okay, so I will open his slides and will leave the room for him and just ask him to come over to my office. Okay.
Now, thank you, Gabriella, for your introduction. Today I'm going to present about my work on purification and characterization of tomato-derived vesicles. So the recent reports that they are indicating that plants also secrete membrane vesicles, and they are also derived from the plants like ginger, lemon. Hi.
This is the voice.
Oh. So here today I'm going to present about plant-derived vesicles. So here in the webinar, most of the people are working on mammalian-derived vesicles. But today I'm going to present about vesicles which are derived from the plants. There is a lot of reports which are saying that plants also secrete nanovesicle-like structures, and different characterization studies provided that these vesicles have structures like mammalian exosome, and they play an important role in therapeutic activity and cell-cell communication. In the slide, you can see that some of the examples for the vesicles derived from the ginger, orange, grape, ginger, grape, and grapefruit. And these sizes you can see in some of the sizes. Also, these TEM images, they are showing they have mammalian exosome-like structures. As I mentioned, that these have therapeutic activity, and they have instinct therapeutic activity towards various diseases like anti-cancer activity, anti-inflammation, and they could be used as a drug delivery vehicles, and they could action on gut microbiota. For example, ginger-derived vesicles have shown the derived vesicles protect the mice from the DSS-induced cancer, and also lemon-derived vesicles have shown the inhibited the tumor growth, all tumor growth. Come to drug delivery application, as I mentioned, these vesicles could be used as a drug delivery systems because of this unique size, structure, stability in GI tract, and low toxicity and the large scale production make them ideal for the drug delivery system. And not only we can use small we can use as a for we can deliver small molecules, nutraceuticals, RNAs, and proteins. For example, ginger derived vesicle could deliver synthetic drugs like palsitoxal, anti-cancer drug, and also ginger derived vesicles could deliver anti-inflammatory drug like mesalazine. Also, for the one interesting thing is they could deliver nutraceuticals like curcumin. We have reports on curcumin, and the nutraceuticals, as you know, as we know that nutraceuticals have high therapeutic activity, but the drug delivery system, development of drug delivery system is challenging for the nutraceuticals because of their low solubility and the low bioavailability. But in the reports, we can see that we could be used as a plant-derived vesicles as a drug delivery system. And come to our lab, we are in a EVMS lab, and we are working on different isolation development of different isolation methods which are efficient, easy to adapt, and for the easy to use and adaptable for the new EV sources. And we are working on isolation of vesicles from the blood and urine. Also, we are working on a production isolation of plant derived vesicles from the different fruits like citrus, cucumber, strawberry, pomegranate, and tomato. And also we are working on the different parts of the plant like leaves, root exudate, and different fruits. And I want to like to mention that most of the isolation protocols developed for the mammalian derived vesicles, but the development of as a protocol for the isolation technique for these plant derived vesicles are quite challenging because from fruit to fruit like citrus, cucumber, strawberry, and they have different properties. So we have to develop a different strategy, and we have to check the different buffers on. After that only we are developing the technique. This is this is our lab group. And now I am working as a postdoc under MSCA grant. My project name is nanoTOM, conversion of plant-derived vesicles into nutraceutical system. Here we want to develop a nutraceutical delivery system for the nutraceuticals, and we have chosen tomato plant as a model plant because this is healthy and most popular in most popular in South Italy, and availability of omic database for the Selenium lycopersicum. First, we want to isolate the vesicle from the root, leaf, and fruit of the tomato. And after successful characterization, we want to use them as a drug delivery system. And in our project, we mentioned quercetin as a nutraceutical for loading experiment. This is my project. And for the today, I'm talking about purification and characterization. For the purification, we need plant material and plant selection of plant. After that, we are going to characterize by physical techniques like TEM, DLS, After that, proteomics characterization for the protein studies, metabolomic studies, and finally we would want to apply the vesicles into biological activity. For the isolation, we have a collaboration with CNR Portici, and there we have plant materials like fruit. We can see the root exudate from the roots of the tomato, and we have tried to isolate the vesicle from the leaves and also from the fruit. And here we have selected two tomato variants for the isolation. One is red sector and another is microTOM. After bring the sample from the Portici, we have for the today I'm going to talk about for the isolation of vesicles from the fruit. So fruits were washed and we make a juice, a homogeneous mixture. You can see in the picture this is a homogeneous mixture, and you can see that isolation protocol in the isolation protocol and step-by-step isolation protocol, and so differential centrifugation followed by differential centrifugation, and is the most commonly used isolation technique for the plant-derived vesicle. And here also we followed the differential ultracentrifugation. In the first step, after homogenization of tomato juice, we subjected differential ultracentrifugation. At 15,000 G, we isolated microvesicles pellet, and after 100 kg, we isolated nanovesicle fractions. After isolation of these nano and micro fraction, we have quantified by using BCA method. After that, for the further application, after isolation, for the further application of biological studies or proteomics, we need more pure vesicles. So for the, and also myself also suggested to do the not only ultracentrifugation, you could another technique, you could combine another technique to purify the vesicles. Here we have used two techniques. One is gradient ultracentrifugation and another one is size exclusion chromatography. Size exclusion chromatography. Here you can see the size exclusion chromatography. The gradient ultracentrifugation was performed by using a continuous sucrose gradient from 15 to 60%. And after the sample was loaded into the gradient centrifuge tube, after that sample was ultracentrifuged at 3 hours for 4 degree. And after, you can see from the slide, there is a separation of, we separated the subpopulation of nanovesicles based on their density, and you can see here the bands with the different densities. And after that, we have collected the fractions to measure the protein quantity, and also we have performed SDS page to separate the proteins and to see the protein profile of the fractions. From the SDS page, we can see that these fractions have different protein patterns. This is from the gradient ultracentrifugation. And another technique we have used is size exclusion chromatography for the purification of vesicles. And the size exclusion chromatography, we use a column with sulfurose 10 ml gradient column with the sulfurose Cl2b. After loading the fractions, we have collected 30 fractions of 0.5 ml 30 fractions, and we found that the fractions were 5, 6, and 7 have protein content, high protein quantity. And to check the different proteins profile, we have used SDS page. From the SDS page, some of the fractions are a similar profile, but some are different profile when compared to the nanovesicles. For the characterization, we used different techniques like DLS, TEM, and AFM, and NCS1, which is based on RPS. From the SDS phase profile, we can see that we could see that we have selected two variants, microTOM and red setter. We can see these two bands which is indicating microvesicles of microvesicles and red setter, microvesicles of two variants. The HDS profile looks different for the nanovesicles and the microvesicles, also different within the between the varieties like microTOM and red setter. And we have used DLS for the size determination, and here you can see the in DLS spectra number of overlaid spectra. But for the crude nanovesicles, we have yellow and green indicates the crude nanovesicles, and their size is 134 and 117 nanometers. And the remaining spectra below 100, we can see from the DLS spectra, these are the sec purified fractions of the micro and nanovesicles, and sec purified fractions of the micro and nanovesicles. And after passing through the sec, we have observed that these have the lesser size compared to the crude and nanovesicles. And for the determination of concentration, we have used two techniques, one NTA and NCS1 instrument. And from the here we have calculated the number of particles given derived from 1 kg of tomatoes, and this is one technique. And from the morphological image characterization, we used TREM and AFM. For AFM, from the TEM images, we can see these are the microvesicles and nanovesicles. They are heterogeneous, and they have the membraneless structure, and this indicates they are structure. And another thing, another interesting thing, we have preliminary data from the AFM atomic force spectrometry, and then these you can see the images of tomato microvesicles of tomato, and they are also like membranes. They are also like spherical, and they look like spherical. And from this micro, we can conclude this the structure of the micro and nanovesicles. After the isolation characterization of tomato vesicles, we did proteomics in gel in solution based proteomics to identify the number of proteins present in different vesicles. Most fractions we found that 900 common proteins in micro and nanovesicle fractions. And in the table, I have summarized top 10-20 proteins in the micro and nanovesicle fractions. And here we can see the most of the some proteins like heat shock protein, clathrin heavy chain protein, pectin exterases, and ATPases in both in the micro and nanovesicles. And this concludes that these proteins, presence of these proteins in also be found in citrus derived vesicles. And this concludes that these are plant-derived vesicles, and these proteins have high EBPDA score. For the bioactivity studies, we have studied in a C. elegans model for the spinal muscular atrophy in C. elegans models. When we used the nano and microvesicles, nanovesicles shown considerably rescue in dying neurons. And conclusions, into conclusions, we have isolated tomato-derived nanovesicles and characterized them by using different techniques. And tomatoes are valuable natural sources for the both micro and nanovesicle sources. The yields vesicles in tomatoes are considerably higher than the EVs derived from the mammalian cell lines. And the size exclusion method, which is used in our after ultracentrifugation, we have used size exclusion chromatography. When compared to the gradient ultracentrifugation, the size exclusion chromatography is faster and provided more yields comparing with these techniques. And thermo-derived vesicles have shown risk in against SME disorder in C. elegans model. And in the future work, we want to check the biological activity of these vesicles in different cell lines. And also we want to develop a nutraceutical drug delivery system for the nutraceuticals based on tomato-derived vesicles. And I would like to thank you for the group at IBB CNR Portici, Stefania Grillo, Giorgia Batelle, Alessandra for providing plant materials, and IBF Palermo, Mauro, and Samile for the DLS and AFM studies, and C. elegans group in CNR Napoli, Elia Di Scavi and Pamela Santanicola for the C. elegans studies, and mass spectrometry group at Budapest, Hungary, Lilia Taruk, and Sven for the anesthesia for the NCS1 results. And also thank you my group for the support of my work, and thank you for your attention.
Thank you, Ramesh, for this very quick overview on nanoTOM project. And I just try to see if there is any question from the audience. So let's see here. Dashboard questions. Somewhere here, okay. Chats. So if you have any question, can you please just place it in. Okay, there is one from Elias Chiavi, and he would like to know whether you can explain how you listed the protein from proteomics. Maybe you can.
For the proteomics, we have digested the sample.
You listed in the table.
Based on the log probe score, from higher to lower, whatever the top 20 with a higher log probe score.
It's not you are not listening back because it's just written, so you can explain it more in detail if you want. That was you answered. That was okay. Okay, then you can move because we have one computer. Okay, so because we have one computer, then we move on and back with Ramesh. But it's so yes, well, for Elia, yes, Ramesh was saying that the proteins were listed by log probe scores, so they were the most highly ranked proteins in this very small list. Actually, he was saying that there were more than 900 proteins identified in each samples, and these proteins were the 20 top ranking proteins on the in one of the sample. It was just I think an example of for that one. This is Elia. And then there is another question. I will read it. I don't know, maybe you can also see it, but for Ramesh, it's Abram who thanks Ramesh for the nice presentation, and he has 3 questions. So maybe you can see it also, but I will read them. So the first is, as you mentioned, as the first step, in the first step, you made a juice, and the question is, Did you peel the tomatoes before this step, or you squeeze them with the peels? This is the first question.
Yes, we peeled the tomatoes. We removed the exocrop of pericrop of the tomatoes. Then we took the tomatoes for the mixer jar and did in prepare the juice.
Can you read the second?
And second, did you use different sources of tomatoes? Yes, we have used different sources of tomatoes like red setter, microtome, and also some of the tomatoes available from the market also. But for the we use for but I mentioned the results only for the red setter, because in another tomatoes like microtome we have extracted very, very less, and we used them as a to develop the method, but we used different sources. If you, as you mentioned in the beginning of your presenting, different plants were used like nutrient also. So those tomato have higher.
Can you read it loudly in a way that every can?
So does tomato have a higher or similar or lower yield compared to the other EVs from the ginger and turnip? Yes, good question. But when we compare to the other sources, we compared for the sources which we are doing in our lab, strawberry, pomegranate, and cucumber and citrus. And compared to in these four, we found that strawberry derived vesicles have higher yield, and then the tomato derived vesicle, the vesicles derived from the tomato is the next. So, but we didn't compare with the ginger and lemon. Another question from Mauro Mano. Which loading strategies are you currently planning concerning both the methods and the drugs? We are at for this question we are at we want to do loading of drugs, but we are at the starting of stage on this loading experiments, but there are two methods are following passive loading and active loading, and some of them drugs were loaded by, suppose I know one literature from the literature, curcumin loading. They loaded the curcumin into the ginger derived vesicles by using the ultracentrifugation technique. So that and another question from Veronica Carl Iggy. Thank you, Veronica. And dear, so we do you observe uptake of tomato vesicles by some type of cells? Yeah, now we are doing the biological activities in the C. elegans model only, but we want to see this activity of these vesicles on different cells, but we looking for the collaboration for the this work. And Aurora asking, it seems to me that proteins identified in your EV samples were mostly enzymes. Were you expecting such a results? This And this for my in citrus derived vesicles, we have found different enzymes in citrus derived vesicles, but I'm doing these proteomic studies. And how did you isolate the proteins for a sorghum analysis? We used tryptic digestion for the isolation of proteins from the tomato vesicles. And another question from Anna Paulina Ramos. How did you test the vesicles in C. elegans model? For the C. elegans experiments, we are feeding the C. elegans with tomato vesicles, and after feeding, we checked the neurons in C. elegans model to check the bioactivity. And another question from Samuel. By considering proteomics and literature, have you noticed any difference between mammalian and mammalian cells? Yes, there is some difference between mammalian. We observed some differences between mammalian and plant cells. Yes. Now I'm going to give to Gabriela.
So first of all, thank you for all of you for the huge number of questions, very, very interesting ones. And also thank you, Ramesh, for answering most of the questions. Yes, by constant literature, let's say if there is something here, Gabriela and Ramesh. We are very, it's Veronica is saying, and I would like just to read it. I'm not sure that you can see it or not. She's saying that we are happy to see you. Did you observe uptake of tomato vesicles by some type of cells? Yes, not yet. We as Ramesh said before, actually we are just planning to do the cell-based experiments, and looking for collaboration. So this is also, I think, a platform. So if any of you who are with us or listening are interested to do some research on the cellular uptake and the biological activity on these vesicles, we are really very happy to discuss it in details. Let's see if some other, yeah, I think. Maybe it's finished by consenting. Oh no, I think we finished all the questions. Ramesh is smiling. So actually, with this one, I think I would like to say many thanks for your participation, and would see you next week when Claudia Tortillone is going to talk about how EVs are involved in cell-cell communication. So I would like to say again many thanks for all of you, and many thanks to Ramesh. Thank you. Bye-bye.